Good morning, students. Today's topic is transduction. Right now we are studying genetic recombination in bacteria, and in that series we will study transduction. Here again the same person, leader, is associated with a study of transduction in Salmonella. What is transduction? What happens? Here, the genetic material of bacteria is transferred from one bacteria to another by a phage particle, by a virus. It's called transduction. So, to study transduction, we will have to study first the infection of phage in bacteria or infection by virus in bacteria. So, bacterial infection, by phage particle may result into either lysogenic cycle, and that may be a lytic cycle. What is lysogenic cycle and what is lytic cycle? Once when the genetic material of viruses is transferred into bacterial cytoplasm. Bacterial cytoplasm have a genetic material. So, if the phage DNA get incorporated into the main bacterial genome, the structure is called episome. And the cell divides into two. In the same way, the phage particle also doubles and they remain integrated with the main genome. So such cycle, here, it will result into a bacterial cell which has a genetic material such cell will keep on division, and from one they become. So, generation after generation, the phage DNA remain attached with the genome, bacterial genome, and they keep on dividing, and the phage DNA remain along with the main genome. Such cycle is called lysogenic cycle. But in lytic cycle, what happens? If the genetic material dissociates itself from bacterial genome and the the viral genome gets separated from the main genome. This is the first step in lytic cycle from lipogenesis, and they starts producing phage protein and phage DNA. In next phase, the bacterial cell will start producing phage particles. And phage DNA. Then the phage protein phage DNA is packed into phage protein. Packaging of phage particle occurs. Then the cell get lysed and the phage particle get liberated from the cell. So, packaging of genome, phage genome into phage particle occurs during maturation, and sometimes mistakenly, by mistake, some disintegrated broken bacterial genome may also get packed into phage particle, and that phage particle may infect another bacterial cell and can transfer the bacterial DNA from one bacterial cell to another bacterial cell. This is called transduction in bacteria. That means transfer of bacterial genome by phage particle is called transduction. So generally phage DNA is packed into phage protein, a structural protein, but accidentally bacterial genome may get packed, and this is called transduction. Now there are two types of transduction. The first is called general or simple transduction. Simple transduction. Or general transduction. General transduction. In this transduction, any part of the main bacterial genome may get packed into the, because prior to the maturation phase, the phage DNA produces enzyme which breaks the bacterial genome in pieces. So from that piece, any fragment, if get packed with the phage particle, it is called general transduction. But what happens when the phage particle dissociates from the main genome? Some of the DNA length of bacteria also get detached with the phage DNA. That means this phage DNA may contain some bacterial genome, bacterial DNA. So such transduction in which the transferred genetic material is generally associated with phased DNA during incorporation in main genome is called specialized transduction. Specialized transduction. अगर ट्रांसफर होने वाला जेनेटिक मटेरियल बैक्टीरियल जीनोम का वह हिस्सा है जो कि फेज डीएनए से जुड़ा हुआ था ड्यूरिंग इनकॉर्पोरेशन विद मेन जीनोम, तब वहां से जो टूटता है तो वहां फेज पार्टिकल से सटा हुआ डीएनए सेगमेंट ही अगर ट्रांसफर होता है तो that is called a specialized transduction. If any of the DNA particle, not only those DNA segment which was closer to the phase DNA on both sides, if the bacterial genome get detached with the phage particle, जब डिटैचमेंट शुरू होता है तो उसी के साथ अगर कोई हिस्सा चला जाए और तब ट्रांसफर हो जाए फेज के माध्यम से, then it is called a specialized transfer. लेकिन पैकिंग के दौरान अगर कोई हिस्सा टूटा हुआ बाय चांस पैक हो जाए अचानक से बाय मिस्टेक, then it is called generalized transduction. Number 1. So, any bacterial cell in lysogenic phase can enter lytic cycle. There are several stimulators which stimulate a cell to shift from lysogenic cycle to lytic cycle, but that has to be studied more. Now I focus once again on repeating transduction in bacteria. So, if you have to understand transduction in detail, then you will have to study the lytic and lysogenic cycle. What is lytic cycle? Lytic cycle is the infection by virus in bacterial cell in which the phage DNA utilize the cell machinery to produce phage protein and DNA in large amount, and they get packed inside, the genetic material get packed inside protein, and the phage particle, the newly developed phage particle, gets released into the environment by breaking down the cell. That process is called lysis, and that's why this cycle is called lytic cycle. For lytic cycle, it is necessary that the phage DNA will detach itself from main genome if the cell is in lysogenic cycle. And if the cell is fresh, the fresh infection occurs, then the cell which will display lytic cycle will not dissociate its genetic material with the main genome. They will remain in the cytoplasm and will transcribe those protein and enzymes which will seize the bacterial cellular machinery. Then they start producing phage protein and copy of phage DNA in large number. They occupy the entire cell environment. Packaging of phage particle occurs. Newly developed phage particle break the cell wall by enzymes and get liberated into the environment. So this method of transfer of genetic material by virus is called transduction. For transduction, lytic cycle is necessary. There are two types of transduction. If the transfer of genetic material occurs accidentally during packaging of phage DNA into the phage particle, by mistake some bacterial part can be transferred. Then it is called general transduction, simple transduction. But the particle which has to be transferred is those part of DNA, main genome, which was associated with the phage DNA directly on either side, either this side or that side. So, when such DNA length gets transferred, which was associated with phage DNA in main genome, is called a specialized transduction. So, I think this chapter, this topic should be clear before you that what is transduction. Transduction is the transfer of bacterial genome from one bacterial cell to another via phage particle. It was studied by Liederberg in Salmonella and Escherichia coli. Now, how does transduction occur? Transduction occurs by lytic cycle. What is lytic cycle? Lytic cycle is the phase of infection of phage particle into bacterial cell, in which the phage particle reproduces by over functioning the bacterial cellular machinery, by hijacking the bacterial metabolic processes. They produces large number of phage protein, structural and physiological, and enzymes, and produces large number of phage DNA. Then their packing occurs and reproduction of viruses completed by breakdown of the cell wall and release of phage into the environment. So, make a note over lysogenic cycle and lytic cycle. What is the difference between lysogenic cycle and lytic cycle? What are the steps of lysogenic and lytic cycle? Then, what is transduction? What are its types? What are the differences between general transduction and a specialized transduction? General transduction. पैकेजिंग के दौरान गलती से बैक्टीरियल जीनोम का कोई हिस्सा अगर चला जाए फेज पार्टिकल में, then it is called general transduction. लेकिन बैक्टीरियल पार्टिकल जीनोम पार्टिकल का जो ट्रांसफर हो रहा है, अगर वह वही हिस्सा है जो कि मेन जीनोम से फेज पार्टिकल के साथ जुड़ा हुआ था और डिटैचमेंट के दौरान कुछ पार्ट फेज डीएनए से बैक्टीरियल जीनोम का भी जुड़ा रह गया, उसी के साथ चला गया, तो इस तरह का ट्रांसफर स्पेशलाइज्ड ट्रांसडक्शन कहलाता है. दैट इज कॉल्ड स्पेशलाइज्ड ट्रांसडक्शन. एंड विद ट्रांसडक्शन इन बैक्टीरिया our chapter or topic Monera is completed. So, in this section, in this chapter, you have to study the Monera as a kingdom, characteristics of archaeobacteria and eubacteria, major differences between gram-positive and gram-negative bacteria. So, gram staining technique is necessary. the difference between the cellular envelope of gram-positive and gram-negative cells, various types of bacteria. Now, nutrition in bacteria, then reproduction in bacteria. So these were the general processes which we have to study about bacteria. With this, this chapter is complete. One more thing, the economic importance of bacteria. You have to prepare a list of all those bacteria which have economic importances in our life. We can make a list of them. For example, there is a large fauna, unicellular life forms present in our intestine, which helps us in the process of digestion and other mechanisms also. For example, Escherichia coli is present in the intestine, Giardia campans, the protozoa. So, not only harmful bacteria in the environment, the bacteria is useful, helpful for us also. The symbiotic bacteria in leguminous plant fixes nitrogen for us. We get several types of chemicals, isolate chemicals by using fermentation of bacteria. We obtain acetic acid from bacteria. So, making of curd is possible due to enzymatic action of bacteria, due to the fermentation process carried by bacteria. Bacteria is used in factories for several processes for isolation of fiber, fermentation process for making beverages. So, in medicine also we have uses of bacteria for making of antibiotics, penicillin, streptomycin, erythrocin. There are so many antibiotics which we obtain from bacteria. So, another thing is water treatment plant utilizes microbes for purification of water, in which we have oxygenic and anoxygenic filtration of water. So, in every aspect of life, we have microbes and bacteria with their useful products. So, economic importance of bacteria is also a very good topic which you have to prepare. List of beneficial bacteria in different sectors. So with the transduction, I think the topic Monera is clear. In next class we will study about protist.